All right, well, thank you everyone for coming this morning virtually. So as Kurt mentioned, this is actually my 6th year at SIU, and what I'll present today is essentially a culmination of the last 4 years of research in his lab, both as an undergraduate and as a graduate student. And so it's primarily been focused on phylogenetics, so DNA, evolutionary biology, using a lot of DNA data and then incorporating biogeographic analyses into the genus that I spent the most time on, called Euthamia, or they're called flat-topped goldenrods. And so with that, I'd like to bring up my 3 main components of my thesis in a minute. So the group that I'll be presenting on today is called the Astereae. This is the 2nd largest tribe and by far the largest eudicot family, the sunflower family. You see them all over the place. There's tons of daisies blooming right now outside. In the fall, there's tons of goldenrods and asters that are blooming all over the place. They're very ecologically important, essentially wherever they are, and they often comprise a substantial amount of the early to mid-successional communities, particularly in grassland habitats. Most of the taxa are herbaceous throughout North America, especially in the eastern half, but you see a lot more shrubs or subshrubs in the western US, where it's usually a lot drier, much higher elevation. So the 3 components that I'll go over today are phylogenetics. This is DNA systematics over it's called the North American Astereae. It's a clade within that tribe of the sunflower family, and it's pretty diverse florally. We have a couple representatives below here. Then we'll focus on phylogenetics of just this one genus, Euthamia, that I've spent the most time on. And we'll wrap it up with biogeographic analyses of each of the species of Euthamia that we helped determine based on largely molecular phylogenetics, morphological information largely from literature, and then notes about each species range in different publications. So as mentioned, we have essentially a few large groups of interest. There's the Astereae, and within that, there's one really big clade called the North American Astereae. Most of these are endemics to North America. A few, of course, get into Central America or the Caribbean or things like that, but predominantly they're here. And one of the more conspicuous subtribes is the Solidago subtribe, the Solidagininae, in the broad sense. This includes a lot of goldenrods, rabbit brushes out west. They're often known as DYC because they're difficulty in identifying them to species, often throughout the US. And within this broad sense of the Solidago subtribe, there's the strict sense of the Solidago subtribe that contains the familiar Solidago. These are the goldenrods you see blooming everywhere next to parking lots and in, little ditches and stuff. And there's also this group called the Gutierrezia lineage. I'll just call it G lineage because it's easier to pronounce, and I'll be saying it a whole lot. Now, this G lineage contains the genus Euthamia that I mentioned that we'll focus a lot of the presentation on. And the relationships for this entire setup has largely been based on 2 loci, ITS and ETS. They're part of what's called nuclear ribosomal. So they're not like typical low copy genes. These encode for ribosomal subunits eventually. There are 2 major evolutionary uncertainties I want to address here in my thesis presentation. The first one is whether the Solidago subtribe is actually monophyletic. People have gone back and forth. It's been contentious whether or not the Solidago subtribe in the strict sense and the G lineage actually grew together to form a broad sense Solidago subtribe. And the other is about the species relationships and monophyly of that genus Euthamia that I mentioned earlier, which is predominantly throughout the eastern United States. Now, I'll be presenting 3 different types of data, and to make it a little clear, I'll be using 3 different colors. The blue is for ITS, ETS. These are 2 loci you get from Sanger sequencing or PCR amplification. It's a little bit old school, but still very useful in modern day phylogenetics. And then the red and green below that, those are acquired through next generation sequencing or high throughput sequencing. We use genome skimming in this case to get what's called the cistron. Now, a cistron includes the ITS, ETS loci, and so the topologies are usually very similar between those 2. And then the chloroplast genome we've also analyzed and assembled wholly, and I'll abbreviate that as the plasto throughout this presentation. And there are essentially 3 programs that I've been using to make phylogenetics. There's maximum parsimony method, which is also a little more old school, and then the newer types of analyses in the past, few decade or two are maximum likelihood and Bayesian inference, which have a more complicated way of looking at the sequence data and producing topologies and phylogenies out of that. Now, for the introduction here, most of the literature that I'm basing on has been done using just that blue, the ITS-ETS loci, and just with parsimony analyses. And so in the top left corner here, I have MP in the blue box. That's ITS-ETS using maximum parsimony methods. And so these are how the Solidago subtribe and the broad sense sort of related to everything else. Those dotted lines indicate uncertainty. And so we can see from just ITS-ETS, the entire North American Astereae is pretty poorly resolved. There's a very large polytomy here, where we're not sure how things are related to one another. And so I have sort of three sets of questions for the phylogenetics portion of my thesis, which is what are the relationships within that Gucci-Rezia lineage, and how is the G lineage and Solidago subtribe, how are those related to everything else within this North American Astereae clade? Now, if we're to look a little bit closer at the G lineage and Solidago subtribe in the strict sense respectively, we can see that, again, there's very poor resolution within this particular Gucci-Rezia lineage. And I also want to note here that I'm color coding these in just this spot because these are what have been proposed based solely on morphology as their relationships to be. And so here it's unambiguous that the Gucci-Rezia group, which is a subset of the lineage, is monophyletic. And you can see when you look at the Solidago subtribe in the strict sense, essentially nothing here is monophyletic in terms of these groups. And so we want to try and resolve these lineages and understand what sort of subsections or subgroups we should put these in and how they're related to one another. We also have to address monophyly issues for both of these groups, for the G lineage and the Solidago subtribe in the strict sense. And we also want to know if that subtribe in the broad sense is also monophyletic, whether that means the first two groups are sister to each other or if one's embedded within another. Now, this is for the Euthamia part. I'll be using a lot of these cartoons to try and simplify it a little bit. So again, in the top left, there's the data source and analysis type. In the middle, there's sort of cartoon of the phylogeny from literature, in this case for the introduction, or from the trees that I made in my thesis a bit later on. And I want to note that these red dotted lines, those indicate that there are poorly supported relationships based on the tree I'm presenting in the right. And of course, we then have our reference tree from literature or whatnot on the right side. And so you'll see things look like this. This tree is from my honors thesis in 2017 that I wrote with Dr. Newbig. And we can see here there's a few species that are pretty definitively monophyletic, but how they're related to one another isn't entirely certain. There are two clusters that aren't really well resolved, and there's not strong support for this bottom species called Herdipes being sister to the rest of Euthamia. So there's questions over this genus's monophyly as well. So this comprises our third set of phylogenetics questions, which is if Euthamia is even monophyletic, and we want to find what the species are in this genus, and whether there's hybridization among these species within Euthamia. I'm going to come back to this final question about what the species are of Euthamia later and address it at the very end here. But for right now, for this phylogenetics portion, we're primarily using these different DNA sequence sources to understand how these taxa are related, and then we'll use biogeography and morphology to provide some context as to how these things might be related or what the data might mean in relation to one another. So we have quite a bit of materials and methods to get through. So of course, what we first do is we extract DNA, usually from leaves. So we go to an herbarium specimen, or I'll go out in the field, I'll collect a few plants, I'll press them, or put a little bit in silica. We'll extract it with a modified CTAB protocol. This is just for the ITS-ETS portion. We amplify those two loci through what's called Sanger sequencing, or PCR. And so we have 2 primers go over the locus of interest, we run it on a gel, we send it off for sequencing, and we get these little peaks here. These peaks are what the computer is calling each base pair to be. And so we'll go through this, we'll join up both ends, and we'll try and determine what the actual sequence is, what the actual sequence for each sample should be. From there, once we think that we have all the samples right, we'll align them all, put them next to one another, and we'll go through again and see, do any base pairs look weird or anomalous? And then if they do, we can go back again, look at the raw data, and verify, okay, that actually is just a sequence that differs from the others, or oh, I made a mistake when I was assembling it the first time. And so we have this iterative process we go through to validate that we're using accurate DNA data. Now, for the more complicated next generation of high throughput sequencing protocols, we use genome skimming, and this involves quantifying how much DNA we have in each tube from the sample that we extracted, and running it on a gel to determine if it's high enough quality to sequence. The sequencing process for genome skimming is really complicated, so I won't get into specifics here. Essentially, we send them up to a facility, they send us back data files, and then we edit them essentially all in Genius. In Genius, our first step is to trim out low quality data. So in this box in the bottom right, the light blue is high quality data. The computer is confident that's what the base pair should be. But those dark blue squares are base pairs. The computer is not sure if that's what it should actually be. And so we trim out those what's called low quality data first to make sure we're not putting any erroneous or possibly inaccurate sequences into the assemblies that we do. As I mentioned, we have two large sequences that we work with for the cistron and the chloroplast. We first have to filter our reads so we have just sequences for those 2 loci. And so we assemble these 2 references, and that helps get only sequences for that particular either chloroplast genome, plastome, or that cistron that I talked about. Once we've assembled it fully, we can go out, annotate it, and from here we'll look at it. Okay, so are all the genes there that should be? If there aren't, we can go back again, assemble over that portion, and see, okay, this actually is what it is, or oh, there's a weird indel, there's a weird insertion or deletion that I didn't see the first time. So once we're happy with all of our annotations, and they look pretty good, we think, we then align all of these sequences, and these are really long. These range anywhere from, from 8,000 to 155,000 base pairs. And so we align, we go through, we sort of skim over all the data, and we look for odd spots. So these black lines or black dots, those are base pairs that do not match up with what the consensus is. There's a couple in the middle here that don't match up, everyone else does. We'll go through this again iteratively. We'll make sure that we have the right DNA sequences to analyze. If there's an issue, we go back, we reassemble, and we do this iteratively to make sure that we don't have any oddities in our data before doing our final analyses. We do most of our aligning through this, at least for the plastome and cistron data, through this Cypress supercomputer cluster on this program called Exceed Online. And so this will help us align everything, and then we can use annotations in Genius or with other programs, and we can of course go through again and just make sure everything looks right, more or less. From there, once we're happy with all of our sequences, we can then do phylogenetic tree searches. And so we do these initially in parsimony just to make sure the topology looks like there's not any weirdly long branches anywhere. And if there are, we can go through and check again and again until we're happy with our data. And then we can run some more computationally intensive analyses through maximum likelihood methods or Bayesian inference methods. We also did time calibrations, which I'll get to with the results at the very end to provide some context for everything that I go over today. We only used one secondary calibration point. There are really no good fossils throughout this tribe that we can use. And so we're using a secondary calibration point from a larger family-wide phylogeny that's at the base of the tribe Astereae and its sister Euthamia. So we also, once we run these, we look at the output, and in here there's a box. So you want only the highest likelihood here where my mouse is going. We're at the very top. And so you want to trim out all the generations or all of them that don't have an optimum likelihood value. So we'll trim out all those and then only use those that seem to have plateaued at the best likelihood value that they're going to reach. We then often will combine multiple runs just to make it a little more robust and make sure that one run wasn't anomalous or anything. So for phylogenetics, so everything except the time calibrations, we will be looking at maximum likelihood analyses and Bayesian inference for essentially everything, but the Bayesian inference and maximum likelihood for the ITS-ETS data are so similar that it would be redundant to show them both. So as a reminder, we have 3 sets of questions. Our first 2 here are, what are the relationships within this G lineage and how are they related to everything else in this clade of the North American Astereae? And then whether the 3 groups talked about earlier are actually monophyletic. So this is one of the outputs that we get. This is a maximum likelihood analysis for all of the cistron data that we have. That's nuclear ribosomal. And we see there are a few major clades that pop out. One of them is this very florally diverse, what I call aster-like clade, because they have that general motif in their flowers, and the head is that actual entire structure with little flowers within it that poke out. We also see that there is a distinct clade of these two separate groups, what's called Pentachitene and Ericameria, and these have more or less this yellow flowered, either flat topped or all coming out from one spot. A little look at them with their flowers, and we see this motif, this symplesiomorphic motif repeated in the Solidago subtribe, both in the strict sense and also with the Gutierrezia or G lineage. And I want to note that the main group that we were uncertain was monophyletic is monophyletic, using these cistron data that are nuclear ribosomal. So we see that using these data, that the Gutierrezia lineage and group are both monophyletic. The G lineage is sister to that big aster-like clade that's florally diverse. We see that the Solidago subtribe in the strict sense is monophyletic, but not in the broad sense here. Now, in contrast, if you look at the plastome or chloroplast genome data, we see that these 2 major groups that we're talking about are sister to one another, and they can collectively form the broad sense of the Solidago subtribe. And this larger group collectively is sister to that aster-like clade with several subtribes in it. So according to the plastome data, we do have the Solidago subtribe in the broad sense, both in the strict sense and the G lineage are both monophyletic here, although we see some slight topological variance between the 2 data sets. That essentially will wrap up our discussion of the North American Astereae phylogenetics, although we'll get a bit to the time calibrations at the end. So moving into the Euthamia phylogenetics component, we have our 3 questions for this: whether the genus is actually monophyletic, if hybridization is present within this genus, and what the species in this genus of Euthamia are. So if we add some more samples and have a broader understanding of this genus, this is using a maximum likelihood analysis for those ITS-ETS data that are rather limited in resolution. We see that these 2 clusters still aren't very well defined. There's a few other species that we sort of recognize, at least putatively within this group. And much like earlier information, we're not entirely certain if this bottom species, Euthamia hirtipes, groups with the rest of them because close relatives give that branch a very low bootstrap support. So we don't have a lot of statistical support for that being biologically real. So using these data, we're not entirely certain if Euthamia is monophyletic. There are at least 5 clades that pop out using these data. And we also did notice there are some hybrid samples that we found between 2 species, this green scabra here at the bottom, and the red hirtipes all the way at the bottom. Those 2 have a parapatric range. They overlap a little bit in the Florida Panhandle, and we do see hybrids with highly polymorphic sequences in them, indicating they're maybe F1 or F2 hybrids. So if we look at the cistron data, again, this ITS ETS contained within it and a lot more data to also look at. We see a little different topology. We do get strong support for Euthamia being monophyletic. Herpes is confidently placed as sister to the rest of Euthamia, and there is very high bootstrap support. Look for these data about 95 or up. I also want to note that when I have a little asterisk next to a node here where it branches, that indicates it has 95 to 100% bootstrap support, which says, okay, that's almost certainly real. Biologically, we see a little differentiation here in that these two species, Galatorum of Nova Scotia and this very wide distributed species, Graminifolia, do group together with some poor bootstrap support, but one Graminifolia doesn't quite group with the rest. Instead, it's with these Caroliniana along the coast of eastern United States. We also don't really see that either of these two green species, they're not really, neither is monophyletic using these data. However, one cool thing is we found the same hybrids that we did in ITS ETS data with these next generation or high throughput sequencing data for the cistron. We also found another instance of hybridization between this light blue colored Euthamia remota and this dark green Gymnospermoides. We found a sample that has Graminifolia and Gymnospermoides haplotypes. So it has a nuclear in their diploid. They have one from mom, one from dad, and we found that case for two different species popping up. So here's the first one. The black circles and the gray circles are where there are two separate haplotypes for the same sequence, where we got results back and there were two separate sequences that we could parse out by referencing strictly to each parent. Now, I'm going to show you the results from the Bayesian analysis because they're a little bit different for this upper cluster. We see that all the Graminifolias are together now, but there's not strong support for them being there with Galatorum. The remota samples are sort of all over the place, although Caroliniana groups, it almost groups completely together now using these data. But we still don't see strong support for either Scabra or Gymnospermoides being monophyletic, and we may just not have enough data resolution to parse these out currently, or it might be a case where one species is paraphyletic to another. We just don't have the resolution using these cistron data. And then similarly, we also have two parental haplotypes for each of the hybrids present. So the cistron data, similar to the ITS ETS data, say, okay, Euthamia is definitely monophyletic. There's at least 5, maybe 6 clades within this genus, and we found 2 instances of hybridization with separate parental DNA sequences in each sample. Now, if we look at the plastome data, we get a very different answer. In this, only one species, the only one in the western United States, occidentalis, is monophyletic, and none of the others are confidently monophyletic in this. They're sort of all over the place, and while some groups seem to be grouping together, nothing's monophyletic, or nothing's strictly speaking paraphyletic, and it's because they're sort of all over the place, indicating that there's been a lot of hybridization or chloroplast capture going on between these different species. The Bayesian inference tree has very similar results. It just has better support for some lineages. Also, I'd like to remind everyone to turn off your microphones during the presentation. We'll be taking questions at the end. So, like the cistron data, the plastome data still say that Euthamia is monophyletic. It's very likely from these there is some hybridization going on, but we only have strong support for one species being monophyletic, and the rest are sort of a jumble between one another. That mostly wraps up the phylogenetics component of Euthamia currently, and I'd like to move on to the biogeographic component of it now. So what we did for this was 2 things. We did niche distributions or species distribution modeling, and also we did niche comparisons. So the first one, which is a little bit more complicated, was making these distribution models. This is where you take coordinate points of a species, usually from herbarium specimens or collected samples. You plot those on a map, you thin them out to be very evenly distributed, And then you run models over that to try and figure out what environmental variables are constricting this distribution and get a better idea of where the species actually occurs, even when there haven't been points collected in it. So we used essentially two databases to get information for all of North America. We used what's called BioClim or WorldClim. This gave us a bunch of temperature and precipitation variables. And then soil grids is where we got our soil information. So how much clay or sand or silt or pH, what's in the soil essentially. And these are all at a 1 kilometer resolution, which is a pretty high grade. Now, for the models, I'm going to be mentioning two things, the AUC and OER for these, and these are essentially our statistics or our stats to determine how well these models are doing. The AUC, essentially the commission error rate, is how many false positives you had, whereas the omission error rate is the false negative. What this means is that the AUC is a measure of how much truly unsuitable habitat is estimated as suitable, whereas the OER says how much truly suitable habitat is estimated as unsuitable. It determines this by using a bunch of background points or pseudo-absence points that automatically generates near the distribution of species to determine how well the models are performing. So we have essentially two big questions for the biogeography of Euthamia, which is which niche does each species occupy? And then whether the niches vary between closest relatives or putative sister species. So for this, as I mentioned, we got coordinates. I got these mostly from databases being CERNEC, GBIF, but we also have some undigitized collections, including some at the SAU Herbarium that I used to make these models. We formatted our environmental data, and then we did modeling through Max and using STM Toolbox to optimize what parameters go into the model. For the niche comparisons, we have a similar but coarser environmental data formatting, and then we run that script in R, or RStudio. Now, for the niche or distribution models, I want to point out that having an AUC of 100 is fantastic and 0 is really bad. If you have an OER of 0, it's as good as it's going to get. So you want as high of an AUC and as low of an OER as possible. Think if OER has an O, it looks like a 0. That's what you want from that. So for the 9 taxa that I considered modeling, there's Caroliniana, which is, again, this solely eastern United States coast species. It doesn't really get past the Mississippi River here. It did relatively well with modeling against false positives, but not really great with false negatives here. For the smaller ranges or distributions of each species, the models perform much better. So these are really good test statistic scores for this Galatorum in Nova Scotia only. Now, for graminifolia is very, very broadly distributed. The test statistics were not great. The false negative score was especially really high for this with that OER. But this also had to be rarefied a very large distance, and the species has a few different niches depending on where it is. It's much more open, disturbed habitat near the Great Lakes, but it gets a lot sparser out near the westernmost and southernmost of its distributions. Now, Gymnospermoides is primarily a Great Plains species, but there's also this coastal component to it, and it again is also bound by it's also exclusively east and north, more or less, of the Ohio and Mississippi rivers here. It has two niches. One is, of course, a medium or high quality prairie species, and then below, it's this coastal species in southwestern Texas and southeast Texas and southwestern Louisiana. Now, this species is very similar to Caroliniana, except it always stays much closer to the coast, and it doesn't live in the same habitats necessarily, because the former one is almost exclusively in pine stands, as well as very acidic. And this one is more sort of disturbed habitat with soil that's not quite as acidic. And this one, being so sort of coast-locked, except for bits of South Carolina and Florida, performed really well as well. And this one's bound exclusively eastward of the Alabama River. Now, this is a very common range you see for a lot of Gulf Coast species. This is the Gulf Coast plain that's bound primarily westward of the Alabama River. This is essentially the western half of the Gulf coastal plain, which is where it's distributed, and it performed relatively well. There's a lot of species. There's a lot of species in very distinct lineages that have a distribution like this. Now, the westernmost species, I think, had a little trouble modeling because it didn't have the right environmental variable used. This is strictly a riparian or a marsh species occurring throughout the western United States. It's not just in the lowlands, it's anywhere from sea level to about 7,000 feet elevation. And while the false positive score was really good, the false negative of the OER was very high for this, and I think it's because I didn't have distance to body of water at fine enough resolution or included that variable to model the species. Now, these last two are very small distributions. They're endemic essentially to southern Lake Michigan. This one's Remota. There is some biasing in that it does occur more in the Chicagoland area and southern Wisconsin, but a lot more samples have been collected, and it's more common on the western parts of lower Michigan and northern Indiana. And then likewise, there's this last species, Scabra, that's sister or closest relative to that Great Plains predominant species, Gymnospermoides. This one's also almost essentially bound by the Apalachicola River in the Florida Panhandle and the Mississippi River along Mississippi and Louisiana. More points do occur in the westernmost Florida Panhandle, and so there may have been some collection biases or skewed a little bit too far near Louisiana, as that's where most collections have been in the last few years. So essentially what I took away from this is that the smaller distributions are simpler to model for accents, whereas the large distributions where the niche is a little more complicated and nuanced, they didn't perform quite as well. There's also a much larger area and a more complex niche to try and reconstruct. One way to improve this might be to include landscape layers such as the presence of pine forest for Caroliniana, distance to water, or if there's still a remaining prairie in that area for Gymnospermoides. So with this, essentially all species are in prairies, savannas, or riparian wetlands, essentially anywhere fairly mesic or somewhat hydric or wet, where there's a lot of grasses growing, is where these are very common, especially in the eastern US. Now, for the niche comparisons, I'll be comparing the closest related taxa here. The first two are Remota around the Great Lakes and Caroliniana along the eastern United States coast. And here are the distributions in G space, or geographic space. And so the environmental data sampled for Caroliniana first is all of this big black bias file area, whereas it's just that for Remota. This is comparing the overall niches of each species, and given their allopatric and have disjunct distributions, it's not surprising that they have very distinct overall niches and that they're significantly different. And likewise, if we look at just what's called the niche divergence test rather than niche overlap, this looks more at whether or not a species has diverged in the local environment rather than the entire distribution. The blue points here are in what's called analogous environmental space. So what it does, it takes both species, it finds out sort of what a similar E space is, and then it removes them that are outside of that space or don't exist within the same general environmental area. So some of them in the western distribution of Remota also get cut off, and unsurprisingly, these are also significantly different, and they don't share any real meaningful environmental space with one another. The next one about our possible species pairs or sister taxa are Galatorum and Graminifolia. Now, Graminifolia is all throughout here. Do you remember? It's very wide distributed, and there's a 40-degree longitudinal difference from its eastern and westernmost extents. Galatorum, conversely, is just in this part of Nova Scotia here. And I want to point out the blue squares or circles. Those are for Galatorum, whereas Graminifolia is everything else in this map with those pinkish stars. So if we look at this, the overall niches are not actually that different, and I think this has to do with how these programs handle samples in your coastal areas, because they tend to pick up a very strong signal, whether it's in niche distribution modeling or species distribution modeling, niche ecological niche modeling, or for these niche comparisons. And I think that's why they're not labeled significantly different, because so much of graminifolia does occur all the way down to North Carolina along the coast and goes all the way up to Newfoundland and Labrador, right at the very easternmost part of the map. But interestingly, we see that if you take just the shared space, so essentially just this area that I'm showing here, it says their niches have divergent, they are significantly different, and that may be because it's including a lot more of this sort of landlocked area that gets overshadowed a bit when you have so many samples next to coastal areas for the overlap test. This is a little bit surprising. The next one is, because that Remota species, it's ambiguous who it's sister to, either graminifolia or Carolina. I ran it for both species. This is what it looks like. Of course, gram is very widely distributed, whereas Remota is just this essentially grayish spot. And all the gram samples are, of course, these pinkish stars that are distributed very unevenly, though they're present throughout this entire area that I'm outlining. The Remota are only a small subset of that. Now, it's worth noting that both the overlap and the divergence, so the holistic and the local tests, indicate that they are significantly different. And it's probably because even though these are coastal along freshwater, the environmental conditions along saltwater that seem to bias models or comparisons isn't there for here. And there's a lot more sort of landlocked species that aren't near any lake or anything, based on the distribution points provided. So our last one is this not very well resolved clade of what's called Gymnus bromoides and scabra. These are allopatric, although the model says they overlap a little bit. That's an overextension that's not quite accurate. As you can see, very unevenly distributed. There's only one in Iowa for some bizarre reason, according to records as well, which is kind of odd. And you also have this coastal component here, like how graminifolia and Galatorum also both had a coastal component that were pretty close by. And I think because of this, the niche overlap test for their entire niches is not significantly different. I think largely because you have two coastal areas here that the species inhabit, even though there's all this continental space. However, if we look at the divergence test, so looking just at essentially this part of Texas, Louisiana, up to Georgia, they are allopatric, and the environmental conditions on either side of the Mississippi seem to be relatively different, or seem to be a barrier for a lot of Euthamia to get across. And I think that's why those two coastal areas, just on a small scale, are different enough for these to be significantly different pairs. So I want to note that everything here, the knot is the overall, the NDT is the local essentially. All but two overlap tests are significant from one another. For Galatorum and graminifolia, these are sympatric, they occur together at some points, whereas Gymno and Scabra are completely allopatric, or they get kind of close together. So essentially just rehashing what I've said, these are the distributions of the two species, as I mentioned earlier, and then here's the other pair that for its overall was not significantly different. And I suspect in both of these cases, it's because there's a lot of coastal habitat that's shared, or at least close to one another for both of these instances. I do want to note that for the two overall tests, for the two holistic tests that were insignificant from one another, the background tests that are sort of what you refer to after it's insignificant, those background tests were insignificant for both pairs as well that I analyzed. And this indicates that the similarity between the distributions can be attributed essentially just due to habitat availability. So essentially limited access to coastlines, because it's just sort of on one face. So with this, we see that each species essentially occurs in riparian, wetland, prairie, savanna areas, or meadows, or things like that. And we see that on a local level, everyone's pretty much diverged from one another of their closest living relatives, which is assuring, because not all of these are separates in geographic space. and mentioning there were the two that were insignificant, and I had my explanations for why, given the coastal habitat. So with that, those are the three major components wrapped up, sort of. There's still the question of what we treat the species of Euthamia as. There's 5 or 6 clades according to nuclear ribosomal data, but there's really only 2 clades and one's monophyletic based on plastid. I want to note here that these are gene trees, they are not species trees. This is what the DNA is telling us, and that may not necessarily represent what's biologically accurate due to limited resolution or things like hybridization and chloroplast capture coming into play. So how should we actually treat the species of Euthamia, and why did I choose to accept 9 of them? So we have 3 major species concepts. The biological or reproductive species concept doesn't apply normally to plants, so we are excluding that one for the time being. I do want to point out we have had some morphological analyses excluding hair or pubescence or trichomes or whatever on the leaves and parts of the plant. And we do say that there is some sort of differentiation when you look at floral parts, including number of flowers per head and leaf dimensions. There are 3 species in the field that if you see them, they're immediately distinctive. There's a super thin-leaved one, Caroliniana, that's the pine forest on the eastern coast. There's Leptocephala down here. That one has these pellucid dots. If you hold it up to the light, there's like this big shining light that pops up through a bunch of the leaves. And then the only western species up here in buckets, which Dr. Nickerin very graciously provided from Colorado. These ones tend to have glaucous stems, and they don't form these nice flat top inflorescences that you see the rest of the species form. Now, what about the rest of them? So when I looked at what species and their total variability could overlap somewhat with other species, so if it looked like my continuous gradient, there are 2. There's that Caroliniana, and it's also that Herpes, both along the east coast, but these are not each other's closest relatives by any means. And we see that for essentially all these pairs, where species that some instances look like one another considerably, at least with some features, they are not each other's closest relatives. And so I'm fairly confident in distinguishing these based on a mixture of molecular phylogenetic and morphological features, in addition to distinguishing them based on their biogeography and the niches that they inhabit. So morphologically, the closest species which might form an intermediate continuation, you might want to treat as a broad species, but those are not each other's closest relatives, at least according to nuclear ribosomal data. So there's reason for splitting them that way. And a fair number of the species, or at least the clusters, have been resolved in this way. So a pretty good framework for understanding how Euthamia has diverged from one another. And ecologically, we see that, at least locally, there are always divergences for the closest living relatives of one another. So I want to add a little bit of context to this. I mentioned time calibrations at the very start, and I want to provide a little bit of explanation of how Euthamia probably came to be and why there are more than just 1 or 2 species. So we're going to go back to having the cistron and the plastid data, and I'll have these highlights here to represent Euthamia, that lineage it's a part of, and the Solidigo subtribe in the strict sense. This was done on Beast 2 through Cyprus, and I'm showing the mean crown ages, not the stems here. So cistron data says Euthamia is about 7.5 million years old, with the G lineage and the strict sense Solidigo subtribe being a few million older than that. I'll show this little cartoon to compare both data sets in a minute. Whereas chloroplast genome data, they say that Euthamia is only about 4.5 million years old, with the G lineage and Solidigo subtribe in the strict sense about 1 or 2 million years younger than in the cistron data. This is probably because the relative amount of genetic variability or variance within Euthamia is smaller relatively to everything else in this plastid data set compared to the cistron data set. So this is what plastid shows, then you put them next together. The ones that are dotted are hash marks. Those ones are from the nuclear ribosomal data, cistrons, and the whole lines, those are from the chloroplast genomes or plastomes. And so you see they're actually not terribly dissimilar for a lot of these. And if we look at what was sort of going on geologically during this time period, we see that most of the divergences diversification happened probably, or at least began, in the mid or late Miocene, with Euthamia probably diversifying in the late Miocene or early Pliocene. So during this time, what was happening? So if you go to literature, the big thing happening ecologically, at least in North America, was the expansion of C4 grasslands. C4 is a more efficient form of photosynthesis that many lineages co-opted, especially grasses and sedges. And we saw the divergence of many dominant C4 grasses, at least their lineages or subfamilies or tribes, during the mid-Miocene, and it was greatly expanded. You saw a lot more fossils during the late Miocene, indicating they were colonizing vast swaths of North America, at least during this time. And Euthamia species very largely inhabit C3 or C4 grasslands. That's prairies, savannas, meadows that are mostly throughout eastern North America, whereas Occidentalis doesn't occur quite the same way. It's most just riparian. But living in these different sorts of grasslands may have contributed to Euthamia speciating in the last few million years, and may explain how we got what we have today. So like Gallictorum, that Nova Scotia one, only occurs in meadows in the southwestern half of Nova Scotia. Carolinea is only in pine savannas, and though shares a large part of its range globally with Herdipes, they don't inhabit the same types of grasslands or disturbed areas. And so with this, I've been talking long enough. I'll wrap it up here. So Euthamia is definitively monophyletic based on the next generation sequencing data we have. There's 9 species, and most of those are in eastern North America. I know there's a lot of sympatry and range overlap among these species. There has been local divergence for, at least based on the Cistron and ITS-ETS data, the closest living relatives of one another. And then we see that the Gutierrezia lineage and Solidae and the Strixons are both monophyletic, but it's uncertain whether the subtribe in the broad sense is monophyletic, since we have conflicting Cistron and Plastome data sets. All of these likely diverged in the mid to late Miocene as the C4 grasslands were becoming a lot more prominent in eastern North America, and that's where a lot of them occur today. And so with that, I of course want to thank my committee for all of the help over these 6 years I've been at SIU, including my various other advisors, official and unofficial. Of course, the students in Dr. Newbig's lab, and also Dr. Bruns for helping me with the niche modeling, and all of the many funding agencies that have made this research and degree possible. And so with that, I will be taking any questions you might have or answering what all these pretty flowers in the background are.